We're going to take a look at signal transduction in this lecture. So remember, in signal transduction, transduce means to convert, and we're just going to be converting the original messenger that bound to the receptor to a signal inside the cell that's going to cause the cell response. And there's going to be differences in signal transduction in intracellular receptors and also in the cell surface receptors. So let's start with the intracellular receptors first, because these are probably the easiest. So remember that intracellular receptors are going to be bound by those hydrophobic ligands. And these are going to be located inside the cell, either in the cytoplasm or in the nucleus. And it's important to remember that when these intracellular receptors are bound by their messenger, we're going to be activating genes or either turning them on or off. And this is going to be affecting protein production. Because remember that genes code for proteins. And this is going to be slow onset because it's going to take some time to turn the gene on or off. And this is going to be a long-lasting response in this target cell, because once you make the protein, it's going to last for a while, so it's going to hang around. So here's just a picture showing an intracellular receptor. So we've got our hydrophobic messenger out here, either a steroid or icosanoid messenger. And because they're hydrophobic, they can just diffuse right through the target cell membrane. And then the intracellular receptor will either be in the cytoplasm, and the hydrophobic messenger will bind to it and then it moves into the nucleus, or the intracellular receptor will already be in the nucleus. So it can either be in the cytoplasm or the nucleus. And then it will then bind to the DNA. And this picture is showing we're turning on a gene. And when we turn the gene on, it'll be transcribed. We'll copy it, make the messenger RNA. And then the messenger RNA gets sent out to the cytoplasm, where the ribosomes will hop on, translate the messenger RNA, and then we'll make that protein. And then that protein will cause the cell response in the target cell. So the cell surface receptors are going to be used by the hydrophilic messengers. And remember, these hydrophilic messengers are going to be made of amino acids, the amines, except for the thyroid hormones, and then they could be protein or peptide messengers as well. So these receptors are going to be located on the cell surface in the plasma membrane. And there are 3 different types of cell surface receptors. So the receptor could be an ion channel, the receptor could be an enzyme, or it could be a G protein linked receptor. And these cell surface receptors are generally going to cause a cell response that's going to be fast in onset. Because if you think about maybe the receptor is an ion channel, well, it's going to be pretty fast to open once the messenger binds to it. And we're going to have a short-lived cell response in the target cell because, for example, like that ion channel, It's going to open quickly, but then it's also going to close quickly as well too. So here's just a picture showing the 3 different types of cell surface receptors that can be located in the plasma membrane. So first we've got this channel-linked receptor. So the receptor is just an ion channel. So it's got a gate on it. And when the hydrophilic ligand binds, that'll either open or close the gate. Here we're showing that the gate opens. And these ion channels are going to be very specific for what ion they're going to be letting in or out of the cell. And this one happens to be letting sodium ions into the cell. And then that will cause the cell response. And then next we've got this enzyme-linked receptor, where the receptor is actually an enzyme. And when the hydrophilic ligand binds to the enzyme-linked receptor, that'll turn the enzyme on. And the enzyme will end up phosphorylating proteins. And then those phosphorylated proteins will cause the cell response. And then the third type of cell surface receptors are these G protein linked receptors. So when the hydrophilic messenger binds to the G protein linked receptor, that'll activate this G protein. Part of the G protein will take off. And the G protein can go and do one of 2 things. So it can either go and open or close an ion channel, and that'll let a specific ion in or out of the cell, and then that will cause the cell response. Or the G protein could go over and turn an enzyme on or off, and then that enzyme can make second messengers, and then that would cause the cell response. So the G protein linked receptors can do one of 2 things, either open or close ion channels or turn an enzyme on and make these second messengers. So let's take a look at these channel linked receptors. They're also called fast ligand gated channels. And that's just because they open and close very quickly when the hydrophilic ligand binds to them. And what's going to cause the cell response is when the ion is going in or out of the cell, and that ion can change the membrane potential, or if the ion is calcium, the calcium can cause a couple different things. So let's just take a look at a couple of pictures here. So here we've got one of these channel-linked receptors. The gate in the middle here is closed, but when the hydrophilic messenger binds, we open the gate, and then whatever this channel is specific for, that ion will move either in or out of the cell depending on the ion, and then that will cause the cell response. And then here's an example of a specific channel-linked receptor. This one is going to be bound by a neurotransmitter called acetylcholine. So maybe this is a skeletal muscle cell. And the motor neuron releases the neurotransmitter acetylcholine that'll bind to this, they call it a nicotinic cholinergic receptor here for the acetylcholine. And when acetylcholine binds, it opens the gate, and it's going to let mostly sodium into the skeletal muscle cell. And then sodium will cause the cell response, which will cause the skeletal muscle cell to contract. And we'll learn how that happens later on. And then this is just another example of a channel-linked receptor, just showing the gate closed here. Hydrophilic messenger binds, we open the gate, and this one is specific for calcium ions. Calcium will enter the cell, and then depending on the cell, the calcium can do several different things, and then the calcium will cause the cell response. Okay, another type of cell surface receptor are the enzyme-linked receptors. So the receptor is actually an enzyme. It's going to be turned off, until the hydrophilic ligand binds, and then the enzyme gets turned on. An example of an enzyme-linked receptor is the tyrosine kinase receptor. And these are going to be involved in growth and metabolism responses. So these will be bound, for example, by messengers like insulin and growth factors. And notice that we've got, in our name of our enzyme, we've got kinase. Remember we learned before that kinase enzymes, they phosphorylate things. So they're going to add phosphate groups. So here's an example of an enzyme-linked receptor. So we've got the enzyme here that's in the plasma membrane, and it's turned off right now. But when the hydrophilic messenger binds to the enzyme, it turns this tyrosine kinase enzyme on, and then the enzyme will grab a protein and some ATP. It's going to take a phosphate group off the ATP, and then it's going to phosphorylate this protein. And it specifically will phosphorylate the amino acid tyrosine in the protein, and that's how it got the name tyrosine kinase, because it actually phosphorylates that tyrosine amino acid in the protein. Okay, but just remember it's a kinase enzyme, so it's going to phosphorylate things. And then this phosphorylated protein will then cause the cell response in this target cell. So maybe it's going to turn, some other enzymes on or off. Maybe it'll end up turning genes on and off. So there's a number of things that can happen there. Okay, here's a picture of the insulin receptor. So the insulin receptor actually has two parts to it, and we have binding sites for two insulin molecules here. Insulin is a protein hormone, so it's hydrophilic, and when insulin binds to the insulin receptor, that'll turn on this tyrosine kinase enzyme, and then it'll phosphorylate these proteins, and then these phosphorylated proteins will actually go tell the cell to put out its glucose carrier proteins, and then the cell can then transport glucose into the cell because you just ate and there's lots of glucose out there for the cell to take in. So that'll be the cell response to put out the glucose carrier proteins. Okay, and if you look at this picture, there is something really wrong with this drawing. So what is it? Okay, hopefully you noticed that the phospholipid bilayer is backwards. So you can see that the heads of the phospholipids are turned inwards and the tails are sticking out. That's totally backwards. So remember that the tails should be facing inwards and the heads should be on the outside here, because remember the fatty acid tails, they like to huddle together on the inside of the membrane, and so I don't know why it's drawn like that, but it's all backwards. So